Let's talk about a world that's all around us, on us, and even inside of us, but it's completely invisible. It's a universe just teeming with life, from microscopic creatures in a single drop of water to the very cells that make you who you are. And for almost all of human history, we didn't even know it was there. Today, we're going to explore the incredible story of the tool that gave us the key to see it all, the microscope. So let's just start with a huge question. Imagine you knew there was an entire secret world right under your nose, but you had absolutely no way to see it. How would you even begin to study it? For most of history, this wasn't just a fun thought experiment. It was a genuine reality. I mean, people could see the effects of this world, like getting sick, but they had no idea what was actually causing it. All they could do was guess. But that all changed when a few really curious people decided to stop guessing, and instead, they built a key. That key, of course, was the microscope, and it would completely change how we understand, well, just about everything. So, who were these first visionaries? Let's meet the people who took the very first peek into the invisible and showed the rest of humanity what it had been missing all along. Okay, so it all kicks off around 1590 with a father-son team, the Janssens. They were spectacle makers, and they figured out that if you put two lenses inside a couple of sliding tubes, you could make small things look a lot bigger. But then, in the 1660s and 70s, two absolute legends enter the scene: Robert Hooke over in England and Anthony van Leeuwenhoek in the Netherlands. These guys took that basic idea and just ran with it. And what's so fascinating is that these two pioneers took completely different approaches to the same problem. On one hand, you've got Robert Hooke with his big, complex, kind of fancy-looking compound microscope. And on the other hand, you have Leeuwenhoek, who made these tiny, super simple handheld devices with just one but incredibly powerful lens. Two totally different paths leading to the same hidden world. So picture this. Robert Hooke is looking at a paper-thin slice of cork. Like the kind from a bottle stopper. Under his microscope, he sees that it's made of thousands of tiny, empty boxes. They actually reminded him of the small rooms or cells where monks lived in a monastery. And just like that, he gave us one of the most fundamental words in all of biology. And this right here, this is the moment of discovery. Hooke published all his findings in a book called Micrographia, and it was filled with these mind-blowing drawings of things no one had ever seen before, like the eye of a fly or the structure of a feather. When people saw it, they were just floored. It was like seeing pictures from another planet for the very first time. Now, at the same time, Leeuwenhoek's simple-looking device was an absolute powerhouse. He was a master at grinding lenses, a skill he kept a secret his entire life. And his tiny single lenses could magnify things up to 270 times, which was just completely unheard of back then. He was seeing things with more clarity than literally anyone else on Earth. So what do you see with that kind of power? Well, he took a look at a drop of pond water and saw that it was swarming with tiny creatures swimming all around. He was the first person ever to lay eyes on living bacteria and other single-celled organisms. He called them animalcules, which basically means little animals. And honestly, that's exactly what they looked like to him. But, just seeing things wasn't quite enough. For the microscope to become a real scientific tool, they had to solve a huge problem. The image itself was often a blurry, colorful mess. And this led to the next great challenge in our story. Yeah, here was the issue. While those early microscopes made things bigger, they weren't very clear. Looking through one was like trying to see through a warped, wavy piece of glass. Everything had these weird, rainbow-colored halos around the edges, which made it impossible to see any fine details. This one flaw actually held back scientific progress for more than 100 years. The technical term for this pesky problem is chromatic aberration. What's happening is that a simple glass lens bends different colors of light at slightly different angles. You know how a prism splits white light into a rainbow? It's basically that. It means that the red, green, and blue light don't all focus at the exact same spot, and that's what creates that blurry, color-fringed image. The big breakthrough finally came in 1830 from a man named Joseph Jackson Lister. His idea was just brilliant. He realized that if you combine lenses made from different types of glass, they can actually cancel out each other's color-bending flaws. By carefully placing these different lenses at very specific distances, he created the first achromatic lens system, one that produced a sharp, perfectly clear image. This was the moment the microscope went from a curiosity to a serious scientific instrument. And then, things got even more precise. In
In the late 1800s, a physicist named Ernst Abbe started working with an instrument maker, Carl Zeiss. Instead of just using trial and error, Abbe applied some serious math and physics to the problem. He actually came up with formulas that could calculate the absolute best, sharpest image you could possibly get with light. He pushed the optical microscope to its absolute physical limit.
So the light microscope was basically perfected. Scientists could see cells and bacteria with incredible clarity. But pretty soon, they hit a wall, a fundamental law of physics. They wanted to see things that were even smaller still. And this is where you get to a really mind-bending question. See, light travels in waves, and you simply cannot see an object that is smaller than the wave you are using to look at it. It's kind of like trying to pick up a single grain of sand with a giant bulldozer scoop. It's just not going to work. So the smallest viruses and even molecules, they were still totally invisible. The answer, figured out in the 1930s by Ernst Ruska and Max Knoll, was, well, it was radical. If the waves of light are too big, you just stop using light. Instead, you use something with a much, much smaller wavelength, a beam of electrons. This chart really shows you just how massive of a leap this was. Instead of shooting light through polished glass lenses, an electron microscope shoots a beam of electrons and uses powerful magnets to focus them. And the difference in power is just staggering. A really good light microscope might get you, say, 2,000 times magnification. An electron microscope, it can get you over 1,000,000 times. This wasn't just an improvement. This was like discovering a whole new level of reality. Now, the invention of the electron microscope didn't make the light microscope obsolete. Not at all. It just gave scientists another tool. And today, we have a whole microscopic toolbox with different types of microscopes designed for very specific jobs. Even with electron microscopes, there are different kinds. A transmission electron microscope, or TEM, works a lot like a slide projector. It shoots electrons through a super thin sample to give you a flat 2D picture of what's inside a cell. A scanning electron microscope, or SEM, is different. It bounces electrons off the surface of an object to create these incredible detailed 3D images of its texture. So a simple way to remember it is TEM for the inside, SEM for the outside. And we've even found amazing new ways to use good old-fashioned light. Fluorescence microscopy is a total game changer. Scientists can attach tiny glowing markers to a very specific part of a cell, like one particular protein. Think of it like putting a tiny glow stick on just one player on a football field. When you shine a special light on the cell, only that one part lights up. This lets scientists literally watch a single process happen inside a living cell, in real time. It's amazing. So, from the Janssen's simple little tubes all the way to watching glowing proteins move around in a living cell, the journey has been absolutely incredible. But it is definitely not over. Technology is pushing the boundaries even further. You see, the next big revolution is all about computers. Modern microscopes are hooked up to high-resolution cameras and incredibly powerful software. This lets scientists instantly create 3D models from their images, use software to make things even sharper, and even use AI to automatically find and track cells as they move and change. This combination of optics and computing is just making discovery happen at a breathtaking pace. So, a journey that started with 2 simple glass lenses in a tube has now brought us to a point where we can watch individual atoms do their work in real time. It really makes you wonder, every time we find a new way to see the impossibly small, we discover a whole new layer of reality we never knew existed. So the only question left is, what invisible world will we unlock next?